Okay, next slide. How am I doing this here? Enter isn't working for me. I'm not sure why.
You tried.
Oh, here we go. Here we go. It works. Okay, I also want to acknowledge that I live, work, and play on the Coast Salish territory. And I would also add that this particular topic does have implications for Indigenous communities along the Pacific coast. And so it is an important one to look at. Today, I really want to just have a discussion about marine biotoxins of concern for human illness. I want to review a history of some recent illness events for shellfish, look at climate factors, because I think that's why people are very interested in this topic, but also interlink it with policy issues. And then instead of talking about how we investigate foodborne illness at the beginning, I've decided to switch things up and put that at the end. After we talk a little bit about acute and long-term exposures and how we can improve our investigations. So today's focus will be on paralytic, amnesic, and diarrhetic shellfish poisoning. These are the main shellfish issues in BC. So what you see on your screen right now are pictures of phytoplankton and diatoms. In that picture that you see is Alexandrium. So there are 3 species of Alexandrium found in BC. 2 are linked to harmful algal blooms, and those are Alexandrium acatanella and Alexandrium catella. So in B are phytoplankton called Dinophysis. There are 4 species found in BC, and 1 is linked to harmful algal blooms, and that's Dinophysis acuminata. There's also other phytoplanktons that release these diarrhetic shellfish toxins. They're called Prorocentrum nema, but so far it's only caused a harmful algal bloom in the Arctic. And then there's also other species like Phallochroma. At the bottom, that rather spiky looking thing is diatom, and Pseudonychia are of great concern, although we haven't seen them too much here. It's Pseudonychia australis that causes illness. So when I say these cause illness, these phytoplankton and diatoms, they release toxins. So what about the marine biotoxins that aren't here yet? On the right-hand side, this graphic is from the state of Florida, and it's actually a warning to people about going down to the beach. In Florida, they have harmful algal blooms from a phytoplankton called Pyrenea brevis. And this is a type, there's other types as well that release what's called the biotoxin. And this toxin can actually be aerosolized. And this has been an issue in the southern US of great concern, and it closes beaches, especially to asthmatics. So the paper that's referenced there and this graphic, this causes neurotoxic shellfish poisoning, and we don't get this in BC. But the reason they had this hotline set up was that the Poison Control Center was getting overwhelmed with calls. So they actually set this up as a separate hotline to tell people about this issue. And to be clear, we don't have this issue in BC. Other types of toxins are asparacids and tetrodotoxin, which you may have heard linked to pufferfish. And there actually are many, many other toxins in BC that affect plant and animal health, but I'm going to just focus today on human health. So how do we get exposed to toxins produced by harmful algal blooms? I mentioned already aerosols. The first one, of course, is through food. If we ingest food and we ingest the toxin that's accumulated in the shellfish, but you can also get occasional exposure if you're accidentally ingesting. Most people don't try and swallow seawater, but when you're swimming in it, that might happen. And that's shown that fellow that's swimming towards a red algal bloom. It's probably a good point to say that not all algal blooms are colored. Most of them are not colored, so you can't know just by looking at the color of the water. If it is colored, you can probably say that it might be a harmful algal bloom, but otherwise you don't know. Another good way of looking at how we get exposed to toxic microalgae and toxins in shellfish is looking at routes of exposure in this diagram. And this is a pretty good one. You can see, as mentioned, that it does affect fish and animals and birds, causing fish mortality and shellfish mortality. But I want to draw your attention especially to the right-hand side there, where there's not just bivalve shellfish, but there's also some crabs. So for us here in BC, we might think about Dungeness crabs and other crustaceans and invertebrates. As well, planktivorous fish can accumulate toxic microalgae. So it's something we need to consider in our food chain. So let's do the usual orientation for what each of the three marine biotoxin poisonings are, just in case you're not sure. Paralytic shellfish poisoning is caused by paralytic shellfish toxins, of course, but they're primarily saxitoxins and saxitoxin equivalents. It is of note that there are over 50 different types of these toxins. They block sodium channels in cells. They're voltage sensitive, and the toxin disrupts nerve conduction. That results in paralysis and sensory abnormalities. The symptoms can start very quickly, as you see there under symptoms and onset. Within 5 to 15 minutes, some people have reported they put the shellfish on their tongue and immediately they get tingling or numbness. This can progress to the hands and feet. They may have difficulty breathing. They feel some report a floating sensation or euphoria. They're incoordinated, which has been incorrectly linked sometimes to alcohol exposure. In very serious cases, obviously, if you have too much difficulty breathing, you can go into respiratory arrest, and there has been death. Other symptoms that are also reported include vomiting, especially nausea and vomiting when sitting up, and less often, but still present gastrointestinal symptoms. So at the bottom there, I've got a picture of some mussels and a butter clam. And I just wanted to point out that how biotoxins accumulate in shellfish are not equal. So in mussels will accumulate toxin quite quickly, but they'll also release it or depurate it quite quickly. Whereas in butter clams, there has been evidence to show that once a hot bloom occurs, so with high levels of toxin, if the butter clams accumulate a lot of the toxin, they bind it very tightly, and it can take 1 to 2 years before they release it. So obviously that impacts the ability to harvest these species. The next one I'll talk about is amnesic shellfish poisoning. As mentioned, that's caused by those spiky diatoms, pseudonychia, and they elaborate domoic acid. So domoic acid is a neurotoxin that binds to glutamate receptors. They act on the hippocampus, and they're involved in memory function. These symptoms take longer to develop than paralytic shellfish poisoning. It's usually within 24 hours, there's gastrointestinal symptoms, diarrhea, cramps, nausea, vomiting. But then within 48 hours, other symptoms will develop, such as headache, muscle weakness, and more unusually, memory loss and disorientation. And in very serious or high doses of domoic acid can also cause death. So for you movie buffs out there, it's probable that amnesic shellfish poisoning or domoic acid was the inspiration for the Alfred Hitchcock movie, The Birds, and that came out in 1963. So there was a big bloom of domoic acid in Santa Cruz, California in 1961, and flocks of shearwaters that were eating northern anchovies and shellfish became weird. They started acting very strangely. They were flying into windows and pecking people and vomiting all over the place. So there actually have been a lot of episodes along the California coast described in sea lions and birds, and domoic acid blooms caused by that diatom pseudonychia have caused a lot of problems in animals. The last one to cover is diarrhetic shellfish poisoning. And the toxin here that's elaborated are diaphysis toxins as well as okadaic acids. Now, these inhibit protein phosphatases in cells, and it basically interferes with normal cell function. It affects the sodium permeability, and it makes the cell membranes leaky. which is probably why on the left-hand side of the slide there, you can see that the most prominent symptom for this is watery diarrhea. This can also develop quite quickly. So you can imagine having a meal and within 15 to 30 minutes, you've suddenly got watery diarrhea. Usually the symptoms take 1 to 2 hours to develop, and they almost always occur within 12 hours. The symptoms resolve within a few days, and because they're largely gastrointestinal, it can be really hard to diagnose this illness because there's so many things that cause gastroenteritis. So I want to take a brief look at what's happened over the last 10 years. And since we're focusing on marine biotoxin illnesses today, I just wanted to review this timeline since many other of the outbreaks we've had, the bacterial and viral ones, are also kind of linked to climate change. So in 2011, we had our first diarrhetic shellfish poisoning outbreak, causing illnesses in over 60 people and linked to 15 premises, including restaurants. And that one, because it was such a big cluster, it was unusual, even though it was the first occurrence. And we really haven't seen any cluster since then. Moving along, in 2015, 2016, we had Canada's largest Vibrio outbreak. It caused illnesses in over 80 people from multiple premises. And it's been well described. It occurred during a period of elevated temperature. And so obviously that's climate change related. During that same period, we also had the blob. I don't know if you've heard about the blob. Most people have. It sounds like a sci-fi movie, but it's actually an area of very warm water out in the Pacific. This led to some pseudonychia blooms that might have been linked to a case, but I'll cover that later. Following that, in 2016, 2017, we had Canada's largest ever norovirus outbreak linked to oysters and partially cooked oysters, and this came in 2 waves. 449 people were ill. The next year, we had a non-pathogenic strain of Vibrio cholera causing illness in Indigenous community members after consuming herring eggs. So I just want to say the label of non-pathogenic Vibrio cholera has always confused me, but really this only means that it's not an endemic strain, but it does cause illness in vulnerable groups. Throughout the years, I had to put paralytic shellfish poisoning in somewhere, but throughout the years, we have had PSP clusters. There were some significant illnesses again seen in Indigenous communities. And unfortunately, just last week, we are now involved in another norovirus cluster with oysters. So climate change. This slide is a bit dated. It comes from the Intergovernmental Panel on Climate Change. It just kind of shows what's happening with climate change rise. And I like this slide because the ladies aren't supposed to reveal their age, but I was born in the 1960s. And it kind of freaks me out to look at this and know that during my lifetime, the temperature has gone up over a degree. And we're trying to hold the temperature down, but this is a big issue. So in the context of climate change, I'll continue. And at the link at the bottom of the slide, a report just came out this week from Health Canada or Government of Canada on health in a changing climate. And honestly, I haven't had a chance to look at the report yet, but if you can, please do look at it. It probably links back to this report to show that in temperate areas like we are and in the Arctic, climate change is more rapid than other areas, and it is a big concern for us. So focusing in on PSP, I want to talk a little bit about the work we've done at BC-CBC, and this is from a history of PSP that we published recently. We have also seen links to climate change. So when we looked at all of the PSP investigations over a period of 80 years and compared them to buoy sea surface temperature data, we did see a correlation with increasing temperatures. The number of people ill with PSP per event or cluster has been smaller in recent years, like 1 to 2 to 3 people ill versus in the earlier years, like in the events that we had between 1940 and 1960, that was many, many more people, but fewer events. But every time there's an investigation, it's a burden on public health. So it is a concern for us. looking a bit more at who got sick over this period, this shows that who was ill in these investigations. And in the last 10 years, in the last decade, there were 3.5 times more investigations compared to previous periods. Throughout the period in the blue, you can see that Indigenous harvesters have been affected by these PSP poisonings. But more recently, you can also see recreational harvesters in the lighter blue. And retail and restaurants have shown up in recent decades as well. So this figure is actually showing the same data in a different way. I just really want to emphasize the demographics for these clusters, saying that the majority of the investigations here are self-harvesting incidents. And this is important, I think, from a policy perspective, which I'll get into later. There are events that have been related to commercial product, but overall it's self-harvesting, and the majority in Indigenous harvesters. So almost 1/4, 24% of illnesses there. So where did people get PSP? In the Pacific Coast. This map, again, from the same paper, is showing how often areas around the province were sources of PSP illness, whether they're probable or confirmed. So probably the symbols are a bit hard to see on your screen. The green ones are for Indigenous communities. And if I haven't mentioned it yet, although there were 62 events that we looked at over this period, they do represent over 300 illnesses and 5 deaths. And all 5 deaths occurred in Indigenous people. So it's very important, I think, that we keep track of what's going on with marine biotoxin illnesses. So what were the shellfish eaten? Well, just as you would suspect, the majority of the shellfish are coming from clams of many types, mussels and oysters. One of the issues with the clams being reported is that many people, especially self-harvesters, when they dig up clams and you ask them, well, what kind of clam did you eat? They don't actually know. They just know that it was a clam. And sometimes, for the reasons I mentioned earlier with the mussels and the butter clams and the retention of toxin, some clams will release things earlier and later, and they don't know what kind of species they ate. So they could have been eating a more toxic clam in an area that was partially open. The other thing I wanted to point out are the little orange bars, and those are crabs. So in recent years, we have had PSP poisoning linked to crabs, so not just to traditional bivalve shellfish. So I have here our present and future concerns around marine biotoxins. This is another chart just showing how toxins get into foods. For invertebrates, they include crabs and crustaceans, and they can also concentrate PSTs and other marine biotoxins. And although rare, you can see in the bottom, the PSP illness reports, we have had 3 for crabs. And last year, for the first time ever, we had one in spot prawns. So what do we know about spot prawns? Well, we have had several norovirus illnesses linked to spot prawns, such as the Spot Prawn Festival at Granville Island. And the harvesting season for spot prawns, I think, is around May. But where are they harvested? Well, most of the fisheries for spot prawns are in the Howe Sound area. And these are areas that are often restricted to bivalves because of sanitary closures. So sanitary closures mean that this area might be impacted by a wastewater treatment plant or be near a fishing dock or some other outfall. So bivalves are not allowed to be harvested in those areas, but you may know or not know this, there's no restriction for crabs or spot prawns. And so that is a concern. So what about other toxins? Well, in the graphic on the right-hand side here, you can see that Washington State recalled a whole bunch of Dungeness crabs in January 2021 for domoic acid. So this recall happened just south of us, but we didn't hear anything about this in BC from our regulators, and we didn't know about it. So why is that? I mean, I don't imagine that the crabs magically stopped at the 49th parallel and didn't cross over into Canada. But the real reason is that we don't have any testing program to know about it. And the only report that came actually came through a tribal nation or an indigenous community in the US. So they're much better informed than us. They do have a limit for domoic acid in crab viscera for domoic acid or amnesic shellfish poisoning. So how does Canada measure up on our regulatory? So this is kind of the policy thing here coming up. How do we measure up in terms of our chemical contaminant standards to other countries? By and large, it's pretty much the same across Canada, the US, the UK, and the EU. But of concern, especially when we compare ourselves to the US, they have a definition for domoic acid in all fish that includes other types of seafoods. And they have one specifically for crab, and Canada does not have one. The UK and the EU don't have those standards, but I also don't know if that's a concern in those food groups for them. For PSP, again, the US has a standard in fish. So for them, the definition of fish includes other types of seafoods. And in Canada, for diarrhetic shellfish poisoning, we actually permit or allow more toxin compared to every other jurisdictional district. So these action levels for regulatory action are given in milligrams per kilogram, but the values shown here are the same for parts per million as well. So I wanted to look a bit more about where we can get information about marine biotoxins in our environment. And these charts that are shown here are looking at phytoplankton bloom events. And they're from a very good paper that was written by a collection of DFO scientists very recently that was just published. And it looks at the records of the last 30 years in the harmful algal event database. They call it the HADAT records. And these are recorded blooms from 1987 to present. And on the left-hand side, you can see the Pacific coast, and on the right-hand side are the maritime areas. So this particular chart represents paralytic shellfish toxin blooms. And you can see that over the last 20 years, there have been more harmful algal bloom events reported, but they're consistent in the last 20 years. And there's a lot of them. And I would say that's the same for both the West Coast and the East Coast, that the PST blooms that we're getting are consistent. Taking a quick look at these same kind of records for amnesic shellfish toxins, or this is basically domoic acid. On the West Coast, you can see we have had hardly any. And the first event recorded in 1992 is shown in the star at the top there, but we're getting more and more. So this does appear to be climate change related, or something is changing anyways in the environment. So what does that mean for us? And then on the East Coast, they've had this problem for quite some time. So I mentioned I would talk a little bit about our suspect amnesic shellfish poisoning event that may have involved a case. So this chart that you're looking at here is from our BC CDC maps, and it's pointing out where we thought we may have had very close together in time one event on the right-hand side there that occurred May 21st. That was we would call a probable amnesic shellfish poisoning. So this occurred in a couple that were sailboating at Princess Louisa Inlet. And within a day and a half, one of the sailors had short-term memory loss and could not recall conversations that occurred within the previous 30 to 60 minutes. He was actually flown to St. Paul's Hospital by air ambulance, and they found high blood pressure, heart rhythm irregularities, and no other symptoms. And the physicians diagnosed this as transitory global amnesia, but we couldn't confirm the illness. And there were no samples collected at the site. Again, on the left-hand side, this was another interesting cluster, and I do not think this was amnesic shellfish poisoning. We did have these samples tested, but we didn't find anything. So marine biotoxins can be tricky. Again, these were mussels that were eaten, and there did seem to be some relationship between the symptoms and the weight of the individuals. And I'm going to explore that a bit more later. So where do we have monitoring? The circles here on this chart represent those two suspect clusters. So on the right-hand side at the top there, where there's sort of that little thing sticking out, that's Princess Louise Inlet. There isn't actually biomonitoring there. And all the red dots on this indicate where the biomonitoring stations are along BC's coast. So when I say biomonitoring, this is for marine toxins as part of the Canadian Shellfish Sanitation Program. So they do collect mussels and other kinds of shellfish for testing of biotoxins. So there's a lot of stations there. There's over 80 stations, but the frequency is not always transparent in that often the testing is just before a commercial harvest and it's not longitudinal. And I mean, these tests, collecting samples and testing is very expensive. And I'm not criticizing the program in that way because it's challenging to test everything all the time, everywhere. There's a cost value to this. But in terms of assessing biotoxin risk, it becomes very difficult. The key concern here is that because most of the testing is targeted for commercial reasons, a lot of recreational areas aren't tested or areas where Indigenous communities wish to harvest shellfish. So that can be problematic. Well, going back to domoic acid, this is a picture of a dead whale. And just in the interest of One Health, I wanted to say that although we're looking at illnesses in humans, if you're interested in in the effects that marine biotoxins have on marine mammals, this is a very, very good paper by Lefebvre. It's quite frightening. All of those icons you can see on the right-hand side represent different types of animals, pinnipeds that have died from either PSP or ASP. So I did want to include that. And before the talk, I mentioned I had a couple of videos. So I'm going to skip over these in the interest of time. And if there is time, we'll go back and have a look at them. So this is a little video of a sea lion seizing in 2016 after that warm blob event. And again, related to domoic acid, I have a chart here on the sea surface temperatures. I wanted to go back to kind of to the climate and research issues and just talk a little bit about Alexandrium. They're the ones that, that's the phytoplankton again that causes paralytic shellfish poisoning by releasing PSTs. And the growth of this is more complicated maybe than you imagine. It's not just a passive bit of algae floating along in the water. It takes several forms, including a dormant cyst stage that's in the soils, and it can get churned up. And when it gets churned up, it goes into different stages, and eventually it does become that vegetative algae that can release toxins. So what are the factors that contribute to the development of Alexandrian blooms? That's a pretty big question because that's what's causing the illnesses. So I wanted to take a look back at some work that we did earlier with the help of Vancouver Island University researchers and some of the researchers here in Environmental Health Services. And we did take a look at those marine biotoxin records over a 10-year period. And with random forest modeling, we looked at the toxicity in two different geographic areas, the Strait of Georgia and West Vancouver Island. So the one shown here in cluster A is the Strait of Georgia. And the modeling showed differences, actually. I'll show you West Vancouver Island in a minute, but it showed differences in the importance of factors that led to these biotoxin blooms. So for the Strait of Georgia here, we had sea surface temperature, air temperature, upwelling, salinity, and photosynthetically active radiation, or PAR. In this area, when temperature, though, was even more interesting because it wasn't just increasing sea surface temperature. More blooms happened below 8 Celsius and then above or above 10 to 16. So there was a middle ground where there was really not many blooms. So again, it's complicated. Freshwater discharge does seem pretty important for the mixing of the water column and toxicity events. And I'm really grateful to all the researchers that helped do this work to help us in our understanding. So for the west coast of Vancouver Island, differently from the Strait of Georgia, there were only 3 factors. And believe it or not, temperature was not important here. And maybe that's because it's right by the Pacific Ocean. But again, the factors that were shown here in this modeling graph is upwelling, freshwater discharge, and sea surface salinity. So if you're interested, you can have a look at the papers. So I want to talk about investigating shellfish illnesses. And I want to talk about the initial steps, who gets called first. So for us, poison control, it could be a doctor, it could be emergency, but I want to talk about how the illnesses get investigated and then look at how we can improve the data on our end. So there's a poll question, I think, that should be going up. So if you could please answer the poll. So what we're seeing here is a lot of people are choosing it's eaten raw or cooked. Can people see the poll yet? And some people just say only when eaten raw. Okay, so those of you that answered raw or cooked are correct. You can actually, here's the issue with, oh, am I supposed to press that? Okay, I'm trying to get to the next slide. I'll hit enter again.
You just have to clear their screen and it'll go away.
Okay. All right. There we go. Thank you. Thank you. Sorry for that. Well, the problem with toxins is that they're invisible. You can't taste them. They're odorless. And from a food safety perspective, for someone like me, they're heat stable and they're acid stable. So you can take your crab or your mussels or your oysters and can them. And even the canning won't destroy all of the toxin. They'll still be there and they'll still be in the broth water too. So that's a big issue with biotoxins. So getting back to how we investigate illnesses, this flow chart is from the point of view of poison control. That's who I work with. And on the right-hand side, you can see a form icon. And I want to tell you that there's still always forms involved, and it shows the flow of info from one group to another. So in the first box for poison control, we have some near real-time reports that are generated automatically after a call comes into them. That information gets emailed to us in BCCDC EHS, and we screen it and then send it on to the health authorities, and they do an investigation from their side. But even at the very beginning of this process, poison control, if they see that there is a very serious illness, they will refer it immediately to a medical health officer, and they'll take some steps to determine whether immediate investigation needs to occur in case we need to close a harvest area or do a recall. Following the transfer to that health authority and the environmental health officers that do the inspection, so they'll phone up the case and get extra information. There's another form that gets transferred onto something called the SRI form, the seafood related illness form, and a portion of that form, if it's deemed necessary after another screening by BCCDC EPI folks, that will go to the federal regulators, and they'll look at those forms and collate the numbers over time. And sometimes it'll be necessary, depending on the numbers of illnesses reported, to take actions. So I always take this opportunity when I'm talking about shellfish illnesses to remind everyone of the BC poison control number. It's 24 hours a day, 7 days a week. It's 1-800-567-8911. Really easy to remember, 567-8911. These are the calls that come into us that we do the screening on. And the reason why I really want to point this out is that when I did that review of the 80-year history of PSP in BC, over 60% of all the records came from them, even though they just started keeping records in the 1960s. So that's why in the last 5 years, we've tried to implement this system, or we have implemented this system of this near real-time surveillance. And it's something that I think we can all be really proud of. The reason that we do an initial screening on these surveillance reports is that the way the system is set up is it screens keywords like oysters and prawns and shellfish, but there's a lot of poisonings from oyster mushrooms, for instance. So we don't refer those on to the health authority. We screen out at least 3/4 of the reports that do come into us. In that first flow chart, I talked about key info that people will need to know when they investigate these kinds of illnesses. The key info is where was the shellfish purchased or self-harvested? And that relates back to the harvest area. There's a link there to our shellfish map that interprets the closure data produced by the federal regulators to help people know whether areas are opened or closed. And our poison control folks will often go there to advise people that are out in the field self-harvesting, whether the area is open or closed. So as well as whether the shellfish was purchased or self-harvested, in very serious cases, sometimes you need to find someone who can describe, if the case is unable to, what they actually ate, where they ate it, and again, the harvest area. So I have poll question 2 here. And I was wondering if you could tell me, when we're doing these shellfish investigations, we want to confirm an illness. What's the most important thing to collect? Do we want to collect a sample of the case urine, the case feces, the case blood, leftovers, or all of it? Okay, we've got some results here. We have, some people think urine, some people think feces or blood. Most of you got any leftover shellfish, almost 40%, or all of the above. The actual answer here is not all of the above, it's any leftover shellfish. So the important thing here to learn is that in Canada, we have no lab that can test for the presence of marine toxin in humans. We don't have a lab that'll test for toxin in urine. We don't have a lab that'll test for marine biotoxins in stool or feces or in blood. So the only way to confirm one of these illnesses is actually to collect the leftover shellfish. And these tests are done for us in BC anyways, at a federal lab in Burnaby. They're expensive, just as the biomonitoring program is expensive. They're done on an HPLC instrument. And of course, there's always capacity. And when we have leftovers involved from the case, they'll often test for us on our behalf, which we really appreciate. Okay, so when you normally investigate foodborne illness, and at the bottom is a link to our shellfish-related illness form, by the way, but the EHO or the physician or the public health nurse doing the investigation will get the case contact details. They'll usually ask the person's gender, their symptoms, their food history, what they ate or where they purchased, similar to that key information we talked about earlier. They'll ask about the onset of time to illness, the duration, any occupational or environmental exposures, and any underlying health issues. But to do a good job of shellfish poisonings, it's a poison and the dose makes the poison. And to evaluate the dose, we actually need to ask for information that is not normally requested, such as someone's weight, and then how much did they actually eat? So I have gender highlighted there, but that's because we often, we estimate weight based on gender and age, but it's better to have the actual weight. So the next thing is the amount of shellfish consumed. Did they eat 1 oyster or 10? Did they eat, 1 pound of mussels or 3 pounds of mussels? And then of course, the final thing is the shellfish dose. And hopefully we can get that from the leftovers. But as you know, leftovers are almost never collected during a foodborne illness investigation. So we often look at biomonitored evidence. Briefly, I wanted to talk about a really great bit of work that Mike Lee did for us at BC-CDC, looking at the long-term health effects of some of these marine toxin poisonings. And it's unfair of me to summarize in one single slide, all of the work that he did. But the first thing to know about long-term health effects is that 89.5% of all the literature that Mike found in his review were based on acute events only. And really, we know very, very little about long-term health effects for people that are eating shellfish regularly. The one thing that we can say are the risk groups based on this evidence for each of these marine biotoxin poisonings. And most of the work has been done on amnesic shellfish poisoning in tribal nations in the US based on razor clams. And the concern there is that chronic exposure to shellfish like razor clams that are contaminated with amnesic shellfish poisoning is it actually does have cognitive deficits in people. So their memory and their performance on tasks is reduced just from the food that they eat. So this is a concern. I'm almost done. I wanted to talk a bit about the quality of data that we have for marine biotoxin investigations. So we've got the phytoplankton monitoring that we looked at from the HADAT database. There's some available, but the coverage is poor geographically. Biotoxin monitoring that we have from the federal CSSP program. We do have data, but mainly in commercial areas. So recreational areas and some indigenous areas is poorly covered. Case illness data, we just went through that. Because it's syndromic surveillance, I didn't talk about case definitions, but probable cases are based on symptoms, and we can only confirm in the actual shellfish. So policy improvements, this is where I'm concerned. I'm concerned that we need standards for all fish, especially crustaceans. And I don't know what the answer is, but we need to consider other ways of informing the public risk through either phytoplankton monitoring or creating test strategies that better protect recreational and indigenous harvesting areas. And so the project that was mentioned in the bio, We All Take Care of the Harvest Watch, is a way of empowering indigenous communities to have a look at the phytoplankton in their own areas and help create health adaptation strategies for changing climates. And these are the kinds of things we need to work on. In closing, I just wanted to mention there is another project that I'm working on with Dr. Tom Pisatsky, who's retired, in Mateo, and this chart is from Mateo. One of the policy questions we had here is the threshold for PST sufficient to protect against severe symptoms? So are there any people that are eating shellfish below the regulatory levels of 0.8 mg per kilogram and getting severe illness? And in most cases, that's not the case. And our biomonitoring samples are a good surrogate, and we don't have leftovers, and it shows there is a good correlation. So we're working on that with some researchers from France, and they're going to help us do a more elaborate modeling than what we've shown here. So for my last slide, I'd just like to acknowledge all the people from BC-CDC, including Sarah, who has helped mentor many projects on this for us. Kathleen, who makes all the charts, the poison control, Tom and Mateo and Mike and Aroha and Nicholas, who've all been involved in papers recently on this. And then also the CFIA shellfish desk and the program for all of the testing that they do to keep the shellfish that we have safe, and all of the EHOs that are doing investigations. So that's the end. And I'm happy to entertain some questions.
Thank you very much, Lorraine. Lots and lots of